Today, we are cracking the code of life itself. We're diving into the world of bioinformatics, where biology, data, and pure computing power come together to change, well, pretty much everything. Let's get into it.
So, let me ask you something. How would you read a book that has 3 billion letters and, oh by the way, there are no spaces, no punctuation, and no chapters? That's not a riddle. That is literally the challenge scientists face with the human genome. The amount of data is just staggering, and making sense of it, that's one of the biggest scientific puzzles out there.
So how do you solve a puzzle like that? Well, the answer is this field called bioinformatics. You can think of it as the ultimate decoder ring for the language of life. It's this amazing blend of computer science, math, and statistics, all working together to manage, analyze, and, this is the most important part, actually interpret all that biological data. And here's the thing you've got to remember. Bioinformatics isn't just about storing data in a giant spreadsheet. It's about transformation. It's the powerful toolkit that takes those billions of letters and turns them into actual life-saving knowledge, from understanding diseases to designing new medicines. And today, we're going to go on that exact journey, starting with a single line of code and ending with a potential cure.
All right, our journey kicks off at square one. A scientist in a lab gets a small piece of genetic code. But what on earth is it? What does it do? First, you've got to find a match.
It all starts with something that looks just like this. A simple string of letters, G-A-T-T-A-C-A. To you and me, it's just text. But to a bioinformatician, this short, kind of mysterious sequence could be the key to unlocking a major discovery about a disease.
And that immediately brings up these huge questions. What is this code? Does it belong to a gene we know? And maybe the biggest question of all, has any other scientist anywhere in the world ever seen this exact sequence before? Trying to figure that out by hand? No way. It would be completely impossible.
So, what do? They use a tool that works a lot like a Google search. You know how you type words into a search bar to find a website? Well, a biologist takes their sequence, that G-A-T-T-A-C-A, and plugs it into a program called BLAST. And BLAST then rips through these massive global databases, which contain pretty much every known genetic sequence, to find a match. It's incredible.
But BLAST, is just one tool in the toolbox. It's the go-to for really fast, obvious matches. But what if you're looking for a more distant evolutionary cousin? For that, you might use a tool like FASTA. It's a bit slower, but it's way more sensitive. And then you have the heavy hitters, like HMMR, which use these super advanced statistical models to find connections that other tools would totally miss.
Okay, awesome. We found a match. We've identified our sequence, we know what gene it belongs to. But so what? That just leads to the next big question. What is this gene's actual job? Now it's time to decode the blueprint.
See, a gene is basically a set of instructions. It's a blueprint, and that blueprint is used to build a protein. Now, you can think of proteins as these tiny little molecular machines that are constantly working inside our cells. Each one has a very, very specific job, whether it's carrying oxygen, digesting your lunch, or fighting off a virus.
So that's the next big puzzle, right? If all we have is the blueprint, that sequence of letters, how in the world can we predict what kind of machine it builds and what that machine's job is supposed to be?
This is where bioinformaticians really put on their detective hats. First, they look for homology. It's the classic idea. If it looks like a duck and quacks like a duck, it's probably related to a duck. If our new protein sequence looks a lot like a protein we already understand, they probably do similar jobs. Then they hunt for conserved domains. Think of these like standard issue Lego bricks that show up in tons of different machines and always have the same function. And finally, they zero in on the active site. That's the business end of the protein, the specific spot where all the action happens.
And of course, there's a whole digital toolkit for this detective work. A program called InterProScan is like a master key. It checks our protein for all those common Lego brick domains. Then you have tools like PHEFAM and Panther. They're like these gigantic family tree databases for proteins. They can see which family our new protein belongs to, and from that, we can make a really good guess about its role.
Okay, we are making some serious progress. We know the gene, and we have a pretty solid idea of what it does. But to truly understand how this little machine works, and maybe even how to stop it, we need to see its shape. We have to actually build the machine.
So just remember, everything we have so far is just a one-dimensional string of letters. That's it.
It's like having a complete parts list for a car engine, but absolutely no idea how they all fit together.
So the goal, which is kind of mind-blowing, is to take that flat string of text and computationally predict how it's going to fold itself into a complex, working three-dimensional machine. For decades, this was one of the single hardest problems in all of biology. So how do they do it?
Well, one of the most powerful tricks is called homology modeling, and we can use tools like Swiss model. The logic here is just brilliant. Step 1, you feed it your sequence. Step 2, the software scours a database for a protein that's structurally similar and already has a known 3D shape. Then, for step 3, it uses that known structure as a template, like a scaffold, to build a brand new 3D model of your protein.
And boom! Just like that, you see the true power of bioinformatics. We've gone from a flat string of letters to a dynamic 3D model. Now a scientist can actually spin this thing around on their computer, zoom in on its working parts, and make real, testable predictions about how it works at the atomic level. It's a total game changer.
And this brings us to the grand finale, the part that could literally change lives. Now that we have a 3D model of a protein that's involved in a disease, how do we use that knowledge to design a cure?
Well, the basic idea behind a lot of modern medicine is this classic lock and key analogy. The protein that's causing the problem, that's the lock. A drug molecule, which is usually a pretty small chemical, is the key. And the whole goal is to design a key that fits perfectly into the lock to either jam it up or just change how it works.
But here's the catch, and it's a huge one. There are millions, maybe even billions, of possible molecules that could act as our key. Testing every single one in a real lab? Forget about it. It would take forever and cost an absolute fortune.
And this is where bioinformatics comes in for the win. Using tools like Autodoc, Glide, and Gold, scientists can perform what's called virtual screening. They basically take the 3D model of our protein lock, and then, inside a supercomputer, they test millions of different molecular keys to see which ones fit the best. It's like a massive virtual laboratory running at light speed. So let's just step back and look at the incredible journey we just took. We started with nothing more than a short, anonymous string of letters. We used bioinformatics to identify it, predict its job, build its 3D structure, and finally pinpoint it as a target for a brand new medicine, from raw data to a potential cure, all through the power of code.
And this leaves us with a really powerful final thought. We are generating biological data faster than ever before. This means the next big breakthrough, the cure for a disease we've been fighting for decades, it might not be found in a test tube. It might already be out there, sitting in a database, just waiting in a line of code for the right person and the right algorithm to find it. The only real question is, what will we find next?